G. Wayne Burdell is a third-year Georgia Tech student, getting ready for upcoming final exams. BIO 1520 is his first final, and he is very stressed out because he needs a 95.7 on the final to get an A in the class. In an effort to procrastinate, G. Wayne Burdell tries to figure out how he can sleep better for the few hours he'll be sleeping between now and the end of finals week. He stumbles upon an article
which discusses how a diet rich in prebiotics started early in life can lessen the negative effects of stress on sleep and wake cycles. G. Wayne Burdell has a hard time understanding what this article is about, so he decides to go to the BIO 1520 website and read up on some of the underlying concepts. He learns that prebiotics promote the growth of beneficial bacteria. All organisms can be assigned into one of the three major domains of life: archaea, bacteria, and eukarya. Two of the three domains, domain archaea and domain bacteria, contain prokaryotic organisms, which are single-celled and do not have a membrane-bound nucleus. Domain eukarya contains eukaryotes, which are organisms that have membrane-bound nuclei, as well as organelles such as chloroplast or mitochondria. Species from domain archaea usually live in environments that are too extreme for most organisms. Bacteria, on the other hand, are found almost everywhere, including the land, water, and the atmosphere. They can even be found on our skin, in our mouths, and in our intestines. It is commonly known that some forms of bacteria can cause diseases, but many other forms of bacteria play an important role in our digestive and immune systems. The bacteria that live in our digestive tract have many functions, like helping with the metabolism of digested molecules. These bacteria reside in the large intestine, mainly in the cecum, and communicate with each other through quorum sensing. As the bacteria accumulate to a specific amount, they collectively cause action within the gut. Studies show that exposure to stress can cause a microbial imbalance and disruption of specific gut bacteria, which can have negative impacts on other processes. For example, your sleep-wake cycles. Microbial imbalance can also cause allergic diseases and gut inflammation. One proposed way to combat the negative effects of stress is to consume prebiotics. Prebiotics are substances that can induce the growth and activity of the bacteria within the organism to improve its overall well-being. G. Wayne Burdell now returns to the article and finds researchers hypothesized that consumption of a test diet of prebiotics would reduce negative impacts of stress exposure on sleep-wake cycles and the gut microbiota community when compared to a control diet lacking prebiotics. To begin with, the rats were each randomly assigned a diet, the test diet or the control diet. Both diets had the same nutritional value, but the test diet had significantly higher levels of prebiotics. Each group was then further divided into two more groups based on whether or not they would receive stress. This gave 4 different groups of rats that were being tested: test diet with and without stress, and control diet with and without stress. The stress applied consisted of 100 tail shocks, known to induce a depression and anxiety-like behavior, similar to that experienced after taking a BIO 1520 test. The first major finding was that, prior to stress implementation, the test diet improved non-REM sleep consolidation. This means that rats on the test diet spent less time in wake and more time in non-REM sleep, as described by the following figures from the study, as seen in figures A and B. Figure D shows a direct comparison between diet and non-REM sleep duration. These images are all representative of prenatal days and nights 71 to 72, before stress was applied. 1 week later, the sleep of the rats on the test diet was even more consolidated, as shown. G. Wayne Burdell then read about what happened when stress was applied. REM sleep and non-REM sleep both decreased significantly, maximizing times in wake. A multiple regression analysis was performed to determine a relationship between early life gut bacteria and sleep. All of the bacteria phyla were examined to see whether any individual or combination could help explain why the rats consuming test diet had longer non-REM episode durations prior to stress. This analysis revealed a strong linear relationship between the presence of Bacteria and non-REM episode durations. Overall, this study showed that the test diet, rich in prebiotics, started in early life, resulted in enhanced non-REM sleep due to related changes in a specific phylum of bacteria that could help improve sleep, support gut microbiota, and promote optimal brain psychology and health. The results also showed that the test diet helped with improving REM rebound after stress exposure. The implications of this study show the potential of prebiotics in improving sleep, especially after major stress exposure. As G. Wayne Burdell finishes reading the article, he realizes that maybe eating prebiotics will help him sleep better and even reduce his stress. As he looks at the time, he sees that it is 7:30 a.m. and that he has a final at 8. He scrambles to make it to Boggs in time,
hoping that he can use his newfound knowledge for his next finals experience.